Arrival and preparation phase ensures that every tool and sample is verified before the experiment day begins. Entering the lab at 8 a.m., you start the daily routine by checking the environment for any issues. The security system records your entry at 8:02 a.m., updating the lab log for accurate time tracking. Lab coats are assigned to each scientist, part of the contamination control protocol in the laboratory. Putting on the lab coat is the first step in dressing for the day's experiments and safety. Gloves are essential for handling samples, providing a barrier to prevent any cross-contamination. The snug fit of the gloves ensures dexterity while maintaining safety during all procedures. On the monitor, you review the sample list for today, which includes 12 different specimens to analyze. Each sample has a unique identifier and status, tracked meticulously in the laboratory database. Taking initial notes helps in planning the sequence of experiments and anticipating any challenges. Hand washing is a standard hygiene procedure performed before and after handling any laboratory materials. Proper hand hygiene reduces the risk of introducing contaminants into the experimental setup. Morning interactions with colleagues are part of the lab routine, fostering a collaborative work environment. The experiment protocol binder contains step-by-step instructions for today's procedures, ensuring accuracy. Visual aids in the protocol help in following complex procedures without missing any critical details. The safety checklist has 10 items that must be verified before any experiment can commence. Checking off each item on the safety list confirms that all precautions are in place. Documented safety checks are part of the laboratory's commitment to maintaining a secure working environment. Preparing the first sample tube involves verifying its label and condition before proceeding. With arrival and preparation complete, you are now ready to begin the setup and calibration phase. Re-verifying the sample list ensures that no entry is missed before starting the setup phase, with 15 samples to process today. Each sample status is displayed clearly, allowing for a quick review and confirmation of the day's experimental workload. Writing down key details helps in memorizing the sequence and preparing for any potential issues during the experiments. Detailed notes serve as a personal reference point throughout the experimental procedures, reducing the chance of errors. Collaborating with a colleague on sample verification provides an additional layer of accuracy and shared responsibility in the lab. Gathering calibration tools in advance streamlines the setup process, with items like multimeters ready for use. Proper calibration equipment is essential for ensuring that all measurements during the experiment are accurate and reliable. Calibrating the monitor involves checking brightness and contrast against standard values to maintain visual data integrity. Finding a discrepancy requires immediate recalibration, demonstrating the importance of precision in laboratory work. With all preparations finalized, you are now fully set to begin the setup and calibration phase of the day. The setup and calibration phase involves preparing equipment and systems to ensure accurate experimental results. Preparing tubes requires verifying labels and ensuring they are clean and ready for sample loading, with 20 tubes to process. Accurate labeling on tubes prevents mix-ups and maintains the integrity of the samples throughout the experiment. Pipette calibration is critical for precise liquid handling, ensuring that each measurement is consistent and accurate. Checking the data logging system confirms that it is ready to record real-time data from the experiments. Correcting parameter errors immediately avoids data corruption and ensures the logging system functions properly. The data logging interface is designed to capture all experimental data systematically for later analysis and reporting. Referring back to the protocol binder helps in following the correct sequence and avoiding setup mistakes. Proper lighting calibration is necessary for visual inspections and maintaining consistent experimental conditions. With setup and calibration complete, you are now prepared to execute the experiments according to the protocol. Setting the centrifuge to 3000 revolutions per minute for a 10-minute cycle ensures that samples are uniformly separated, which is essential for accurate experimental outcomes. Cleaning sample bottles thoroughly prevents contamination and maintains the integrity of the substances used in the upcoming experiment. Properly prepared sample bottles are ready to hold experimental materials without introducing errors into the data collection process. Verifying the incubator's temperature stability at 37 degrees Celsius is necessary for maintaining optimal conditions for biological sample incubation. Completing the safety checklist with 5 items ensures that all protective measures are in place before proceeding with the experiment. A thorough safety checklist helps prevent accidents and ensures compliance with strict laboratory protocols during experiments. Labeling tubes accurately with sample IDs and dates avoids confusion and maintains traceability throughout the experimental process. Calibrating the pH meter with buffer solutions ensures precise measurements, which are vital for many chemical experiments in the lab. An organized workbench improves efficiency and reduces the risk of errors during complex experimental procedures and setup. A well-organized laboratory setup facilitates smooth operation and enhances focus on the experimental tasks at hand. Highlighting critical steps in the protocol helps in following the exact sequence required for successful experiment execution. Creating a calibration curve with 5 standard concentrations allows for accurate quantification of sample measurements in the experiment. Saving calibration data electronically ensures it is securely stored and easily accessible for future reference and analysis. Monitoring chemical inventory prevents shortages and ensures that only in-date materials are used in the experiments to avoid errors. Properly stored and labeled chemicals are essential for safe and effective laboratory operations during experimental work. Collaborating with colleagues during setup can identify potential issues and improve the overall experimental design and preparation. A final verification step confirms that all equipment is properly calibrated and ready for the experiment execution phase. Documenting setup completion in the lab log provides a record of preparedness for the upcoming experiments and ensures accountability. With all setup and calibration tasks completed, you are now fully prepared to begin the experiment execution phase according to protocol. Final adjustments ensure that the monitoring systems are optimally positioned for accurate data collection during the experiment. A final verification confirms that all equipment is calibrated and ready for the experiment, with monitors displaying baseline data for accuracy. The prepared laboratory setup features calibrated instruments and organized materials, essential for precise experimental execution and data monitoring. Reviewing the protocol step ensures correct initiation, starting with loading 5 sample tubes as specified in the experimental procedure. Loading samples into the centrifuge requires attention to labels and settings to prevent errors and ensure proper sample processing. Initiating the centrifuge run involves monitoring for 3 minutes to detect any irregularities and ensure stable operation during the experiment. This marks the start of the experiment execution phase, where protocols are followed and data is collected systematically for analysis. Handling centrifuge tubes with care post-run maintains sample integrity for the next analytical steps in the experimental sequence. Sample tubes contain prepared mixtures for analysis, each labeled to ensure accurate identification throughout the experimental process. Transferring precise aliquots to cuvettes allows for accurate absorbance readings, a critical step in the spectrophotometric analysis phase. Running the spectrophotometer scan generates data points for plotting, which will determine concentrations based on the earlier calibration curve. The real-time data graph visualizes absorbance changes, indicating reaction progress with key points marked for detailed note-taking and analysis. Documenting absorbance values in a notebook ensures accurate data capture for later report compilation and experimental validation. Collaborating with a colleague helps verify data interpretation and identify any discrepancies that may require re-evaluation or adjustment. Adding reagents initiates the second reaction phase, ensuring consistent conditions as per the protocol for reliable experimental outcomes. Precise reagent addition and thorough mixing are critical steps that influence the reaction dynamics and final experimental results. Incubating samples at controlled temperature for 30 minutes allows the reaction to proceed under optimal environmental conditions for accuracy. Updating the lab log during incubation keeps the experimental timeline documented and prepares materials for subsequent measurement phases. Preparing the pH meter and cuvettes in advance minimizes delays and ensures efficient data collection once incubation concludes. With 5 minutes left in incubation, reviewing the protocol helps anticipate next actions and maintain smooth experimental progression. Positioning equipment and materials ensures a seamless transition to the next experimental phase after the incubation period completes. Retrieving incubated samples initiates the post-incubation analysis phase, where absorbance measurements are taken to compare with earlier data. Transferring samples to cuvettes ensures accurate placement in the spectrophotometer for reliable post-incubation absorbance readings. The monitor displays dynamic absorbance data, highlighting changes from pre-incubation scans that inform note-taking and analysis. Documenting absorbance values in a notebook captures critical data for later report compilation and experimental validation steps. Calculating concentrations based on absorbance data involves applying the calibration curve to derive quantitative results for each sample. Reviewing calculations with a supervisor ensures accuracy and helps identify any discrepancies that may require re-evaluation. Detailed notes and calculations provide a permanent record for experimental analysis and support future report drafting. Referencing the protocol confirms that all validation steps are followed, maintaining procedural integrity for reliable outcomes. Conducting repeat measurements ensures data reproducibility and highlights any inconsistencies that need addressing. Updating the lab log with new data entries maintains an accurate timeline and prepares information for final reporting. Electronic log updates streamline data management and ensure all experimental details are captured for audit and review. Creating a preliminary report section organizes initial results and facilitates later compilation into the full experimental documentation. Cross-checking notes with a colleague enhances data reliability and helps resolve minor discrepancies through collaborative review. Adjusting equipment settings according to the protocol optimizes conditions for subsequent experimental phases and accurate measurements. Completing the safety checklist ensures all post-incubation procedures are followed, maintaining lab standards and preventing errors. Documenting anomalies and planning reruns addresses data quality issues and aligns with experimental troubleshooting protocols. Cleaning and organizing labware after use prepares materials for future experiments and maintains a tidy workspace. Saving and backing up data files protects experimental results from loss and supports ongoing analysis and reporting. Participating in a team meeting facilitates communication, updates colleagues on findings, and coordinates next steps efficiently. Planning upcoming phases ensures a structured approach to completing the experiment and meeting all protocol requirements. Loading samples into the spectrophotometer initiates the next experimental phase, with real-time data displayed on the monitor for immediate analysis. Recording absorbance values accurately in the notebook ensures that all data points are captured for later review and reporting. The spectrophotometer screen visualizes data trends, providing insights into sample behavior during the experiment. Discussing results with a colleague helps validate findings and identify any potential issues before finalizing the report. Completing the experiment phase involves proper equipment shutdown and material organization, with all actions documented in the lab log. The observation and logging phase begins, focusing on reviewing experimental results and updating all necessary documentation thoroughly. Reviewing collected data on the computer allows for comprehensive analysis and identification of significant trends for the report. The report draft on screen structures the findings, making it easier to compile and present the experimental outcomes clearly. Updating the lab log with final details ensures that all experimental information is accurately recorded for future reference. Comparing notes with the protocol verifies adherence to procedures and corrects any discrepancies before final submission. Printed reports and logs provide a physical backup and facilitate detailed review during the documentation process. Drafting the conclusion section summarizes key findings and their implications, completing the report's narrative structure. Saving and sending the report ensures it is securely stored and ready for supervisor review and approval. Preparing for the next day involves checking the sample list and updating the schedule to ensure continuous workflow. The lab at dusk signifies the end of the day, with everything in order for the next round of experiments. Finalizing the safety checklist confirms that all daily procedures were followed, maintaining lab safety standards. Discussing the day's work with the trainee shares knowledge and reinforces proper documentation practices for future experiments. Backing up files to multiple locations protects data integrity and ensures no loss of critical experimental information. Securing the lab door at the end of the day ensures that all equipment and samples are safely stored until the next use. Clocking out officially ends the workday, with the time logged for record keeping and payroll purposes.